Hello everyone, I'm Eddy Elisée and today I will present my postdoctoral work about identification of diverse amine dehydrogenases by screening biodiversity using sequence and structure-based approaches. This work is carried out between the Laboratory of Biocatalysis, Bioremediation and Synthetic Metabolism and the Laboratory of Bioinformatics Analysis for Genomics and Metabolism from the Genoscope Research Unit Metabolic Genomics. As an introduction, one of the main current pharmaceutical interests is the access to chiral amines, which compose about 40% of the active pharmaceutical ingredients. There are many biocatalytic ways to obtain chiral amines, but our team is mainly interested in native amine dehydrogenases, which I will later call MDH in this presentation. IMDH are NADPH-dependent oxidoreductases which perform the reductive amination of ketone into chiral amines, using ammonia and a nicotinamide cofactor as a hydrid donor. On the MDH4 crystallographic structure on the right, we can see two domains, the N-terminus NADP binding Rossmann fold in blue and the C-terminus catalytic domain in green. About the previous works, after discovering native IMDH through genomic screening of biodiversity, several enzymes were characterized, and some of them were used in organic synthesis. And about 1 year ago, the first metagenomic exploration using MDH protein signature has been achieved. In this context, the MODAMDH project was created with the main objective of finding new MDH enzymes displaying different substrate spectrums and/or structures. However, compared to a classic biodiversity screening using sequence homology to find homologs, here we aim to find distant homologs starting from a large reference family in terms of homology. To do so, we planned a dual strategy. First, a sequence-based approach which aimed to find distant homologs using hidden Markov models, and secondly, a structure-based approach using catalyphores to find active site analogs. Finally, the underlying objective of MODAMDH project is to design a bioinformatics workflow applicable to other enzymatic activities. I will now expose the MODAMDH general workflow. So the first step consists in building two sets of sequences by screening biodiversity using HMM profiles. Next, we clusterize each set to obtain subfamilies of reference MDH in one case and candidate families among NADPH-dependent enzymes in the other case. After this, our selection strategy starts by first generating new HMM profiles for all these new clusters to finally search for distant homologs by screening reference MDH signature against NAD enzyme signature. Secondly, we design catalyphores for reference MDH, which will be searched into crystallographic structures or models from NAD families in order to find active site analogs. Final selection step is the docking of some substrates in the previously found distant homologs and active site analogs to consider them or not as potential candidate enzymes for in vitro analysis such as enzymatic assay or crystallography. The underlying objective here is to iterate this approach by reinjecting the selected candidates into the reference set and restart the workflow. Speaking of biodiversity, we screen several metagenomics databases, including information from diverse environments and from both eukaryotic and prokaryotic kingdoms. On the whole, we considered about 2.6 billion sequences for this work. So now let's start with the selection of NADPH-dependent enzymes. For this step, we decided to use a scope superfamily entry named SSF 51735, which corresponds to NADP binding Rossmann fold domain signatures. This one includes 301 different HMM profiles that we screen against metagenomic databases using HMM search software from HMMer Suite. We performed several filtering steps, including the use of the superfamily script in order to avoid false positives due to inter-superfamily relationships and to check the right presence of a NAD binding domain. With this, we finally obtained a set of more than 20 million NADPH-dependent enzymes. Next step was the clustering of this new set using MMSEX to software in which sequences were clusterized only if the alignment covered 80% of both target and query length. We obtained more than 100,000 families and as many corresponding HMM profiles, and in the end, 104,686 HMM profiles are available for next steps. Now let's continue with the constitution of the reference MDH set. In the previous work, some native MDH were identified and characterized, and the existing family of 2,000 homologous sequences was classified using ISMB method, which classified sequences using structural information of protein pockets, so in this case, active sites. This method combines homology modeling, structural alignment, and hierarchical conceptual classification to finally identify residues correlated to subfamilies' function divergence. Starting from this existing classification, we design HMM profiles based either on each ISMB groups, G1, G2, G3, or 2G8, or on all groups merged together. As a reminder, AMDH structure contains two domains, including an ADP binding domain in N-terminus. So in order to avoid getting hits due to NAD domain signature, we decided to screen metagenomic databases with AMDH HMM profile only based on C-terminus domain. After optimization, we use the AMDH catalytic domain signature built from all ISMB groups, and finally, to be sure, we screen the previous hits with the AMDH NAD domain signature. In the end, we obtain a set of more than 27,000 reference AMDH sequences. Then we clusterize this set using 95% identity and 90% coverage criteria, and we generate the corresponding phylogenetic tree using IQ-tree software. You can see the Cryon group size and in brackets the percentage changes showing evidences of AMDH family expansion with more than 7,000 homologous sequences compared to the 2,000 initial ones. We also clusterized a new set of reference AMDH using ISMB method, and we obtained this graph in which I reported the former ISMB groups using colored labels. Each cluster here is linked to a sequence logo of the corresponding active site, and regarding the labels, we can see also an evidence of AMDH family expansion as we saw in phylogeny. Given that we created the two mentioned sets, we were able to start our candidate selection process. So first, I will talk about the search for distant homologs using HMM comparison. To briefly summarize, we obtained a new set of 27,000 reference MDH sequences from which we achieved a sequence-based and a structure-based analysis. From each, we generated a set of HMM profiles, and we decided to create a global one built from the multiple sequence alignment of the 27,000 sequences. We screened all these HMM profiles against the 100,000 HMM profiles from NAD families, and by analyzing results, the three approaches led to the same family hits, and as a positive control, all the 27,000 reference MDH were found in those family hits. Families containing reference MDH are an access to a large number of MDH homologs that we could analyze, even if the main objective remains to find more distant homologs from a different family. Regarding the family hits, we are currently analyzing them in order to find if there are fragments, what are the domain composition, and do they have already known enzymatic functions. Now, I will introduce the preliminary works about the search of active sites analogs of IMDH. To do so, we use catalophores as 3D signatures of an active site in which you will consider the catalytic amino acids, their function, but also which degrees of freedom you choose for rotamers, amino acid types, etc. For our first screening, we decided to build 3 catalophores based on 3 AMDH crystallographic structures. So as you can see on the picture on the left, and based on mutation experiments, we considered 4 points in our catalophores 3 coming from amino acids, and 1 coming from the NADP cofactor, which is the hydride carrying carbon. The next step will consist in using a fit 3D-based script to find AMDH active site analogs by screening our catalophores against crystallographic structures or models of NADPH-dependent enzymes extracted from publicly available and in-house databases. Also, we will refine our initial catalophores depending on the obtained results, and we will check their sensitivity compared to other oxidoreductase families. Finally, in next steps, the hits resulting from these two analyses will be implied in a docking step with different substrates in order to select final candidate enzymes for in vitro characterizations that will be carried out here at the Genoscope with the help of the bioconversion platform. As a take-home messages, first, the exploration of biodiversity through metagenomic projects is a huge source of sequence novelties with, in this project, more than 100,000 NLP domain-containing families with many spin-off unknown function. Secondly, the combination of sequence-based and 3D-based bioinformatics approaches allows to extend the diversity of enzyme families, but also to find new ones. And finally, bioinformatic predictions and high-throughput screening should gain synergy in an iterative process to learn from experimental results. So to finish, I would like to acknowledge all the people mentioned on this slide who are implied in the MODAMDH project. Also, many thanks to the Agence Nationale de la Recherche for the funding and to the 3D-Sig organizer to let me present this work today. Thank you for your attention.